introduce Hao Chen. So Hao Chen and I have worked together for, I guess, officially almost 4 years now. So Hao Chen is an associate professor at the University of Tennessee Health Science Center, and he got his PhD from the University of Michigan, and he is fully funded, does a lot of
Michigan State.
But I like to describe Hao as the MacGyver of laboratory behaviors and animal behaviors, because you give Hao a Raspberry Pi board and a 3D printer, and he can make just about anything for behavioral testing that you could want. So we could go on and on about Hao and his publications and the work that he's doing, but we're going to let him talk instead of me, and then we'll switch out in the middle of it. So over to you, Hao.
All right, thanks, Laura, for the introduction, and thanks for the chance to discuss the HRDP with you. Okay, so here's the outline of what we try to cover today. So we'll be talking about inbred model organisms, and from that we'll be expanding into recombinant inbreds, and then we'll have a little bit discussion on why some people prefer to use rats in their studies, and then we'll then start talking about hybrid rat diversity panel, which is the main topic of today's webinar. And then Laura is going to discuss the current resources and maybe doing a few demos, and then a few words on where do we go from here. So that's the outline. And the first part is going to be on inbred model organisms. I think all of us know what inbred animals are, but just to get us on the same background. So inbreeding means mating of animals more closely related to each other than the average relationship within the breed, which is the opposite of outbreeding. And within an inbreeding paradigm, you want to pair two animals that are genetically very similar to each other. And the opposite is you want to, in outbreeding paradigm, you want to pair animals that are different or unrelated to each other. So there is a coefficient of inbreeding you can calculate. That is probability that two alleles at any loci are identical by descent. But operationally, inbreeding strains are produced by full sister-brother mating for over 20 generations, and that is also known as full sib mating. So if you generate a pair of breeders, and you just keep full sister and brother mating for over 20 generations, and in the end, the individuals in this population will have virtually identical alleles at any time, at any location on the genome. So that means they're homozygous at virtually all genetic loci. And another property of this population is that they are isogenic, which means all the animals in the same strain are genetically identical. So this does not only include animals from the same generation, it also includes offsprings and like grandparents or grandchildren. Once the population is inbred, each individual is identical genetically, and this can basically be carried on over infinity. So you can basically take the same individual out and study them in different environments, and each individual are basically a clone of all the others. So this is the basic characteristics of inbred animals. With this population of inbreed, there come many features. So one of them is that the phenotypes are stable for each individual inbred strain. This is one of the studies that John Crabb and colleagues did back in 1999, where they were trying to study the phenotypes of 8 inbred strains under 3 different laboratories. They try to standardize all the environmental factors they can think of they standardized on the behavior pipeline, behavior phenotyping pipeline. They standardized even the breeding colony, the housing conditions, the shipping conditions, and the equipment they use to do the tests. Almost anything they can think of, they standardized. And then they did this experiment at 3 different locations. And the results come back with the confirmation that indeed each individual inbred animal, here they have 8 inbred strains, and the phenotypes are stable in 3 different locations. There are other takeaways from this landmark study, but the main takeaway is that indeed with a fixed genome, you can test these individual animals under different locations at different time and different conditions. But under the same condition, you should expect to receive the same kind of data back. So with this stable phenotype, then you can start to think about how to accumulate different phenotypes from the same individual, and then maybe you can maybe, for example, you can study them under different conditions, and you can study a gene by environment interactions, or you can use different type of data and study, for example, phenotype to phenotype correlations, and you can also pile on omic data, and you can study different molecular mechanisms. So one of the advantages of having these inbred strains, of course, is to study gene environment interactions. And that refers to different genomes or different lines of inbred animals respond to environmental changes differently. So here is an illustration of 2 environments, 2 different temperatures, either cold or warm. And each line, the red or the black, represents 1 inbred line. So in this example, on the left side, the red line, the red strain did not have any response to the temperature, but the black did. So this is one of the example of a gene by environmental interaction. And another example is both strains have responded to the environment, but the response is opposite. One goes down, one goes up. And there are other scenarios where there's no gene environment interactions. So this kind of study is better achieved by using inbred strains because you can test the same genome under different environments. One thing that people sometimes think about of inbreds is they tend to expect that within an inbred strain, the between individual variation might be smaller compared to an outbred population, because the assumption is that the genome are identical within the population compared to an outbred population where the genomes are different. That turned out to be not true in many cases. Here is a meta-analysis done by Tudor et al. With Alisa Chesler and Jeffrey Mogul, Vivek Phillips et al. And they collected lots of data from the literature. Basically, they looked at the same kind of phenotyping studies and using both outbred population and inbred population. Here, the inbreds are the open circles and outbreds are the filled circles. And then they looked at the coefficient of variation. And surprisingly, the CV between outbred and inbreds are very similar. There might be many different interpretations for this kind of equal variance between inbred and outbred. One of the hypotheses they proposed is that the heterozygosity in the genome is providing some kind of buffering to the environment stimuli. And in the inbred population, this buffer is gone, so that the individual variation in response to the environment is enlarged. So because of that, you can see that the outbred population and inbred population have a similar coefficient of variance. So with that, we then move on to recombinant inbred panel. So the concept of recombinant inbred panel is not that complicated. You basically take two inbred lines and then you generate an F1. From F1, you can generate F2. If you ignore the crosses over here, you can take these F2s and do a genetic mapping study. What you need to do is to have many individuals, and for each individual, you need to have the phenotype and you need to have the genotype. This has been done many times, and it's a successful strategy to find genetic loci associated with phenotypes. But one shortcoming of this kind of study is that for each individual, you need to generate the individual, and you need to genotype it, and you need to phenotype it. Once you use it, it's gone. So one method that was developed in the 1950s, I think, is to take the F2s and start inbreed them. And by inbreeding, full SIP inbreeding for maybe 20 or 30 generations, then you get individual inbred lines coming out of these two parental strains. So this is the concept of recombinant inbred panel. And here, this diagram is from a review article Carl Broman wrote in 2005. And there are many famous popular recombinant inbred panels, including the BXD panel, many of us are very familiar with. So in the rat world, there's also inbred, recombinant inbred panels. And one of them is this HXB and BXH panel. So in this nomenclature, H is the mother and the B is the father. So the first letter always represents the mother and the second letter always represents the father. So HXB, H here refers to this spontaneous hypertensive rat. The SHR rat is obtained by selective breeding of the WKY rats. And it's being used as one of the parental strains of this panel. The other rat is the B, and this B refers to a BN. So it's a brown Norway rat. But this brown Norway rat is not the standard brown Norway rat that the reference genome is sequenced. This is a brown Norway rat that is a congenic of a particular loci. So this is this RX loci is discovered from an inbred, from an outbred Wista population. This RX loci causes a spontaneous mutation, and with this spontaneous mutation, it comes with a multiple fingers or multiple digits. So that's called polydactyl. And also come with this loci is a dislocated joint. So individual animals with this LX loci have multiple digits, and then their hind leg have dislocated joint, but their forearms are normal, and these animals are still fully active, and in fact, they are very active. So it does not really affect their behavior phenotypes. Most of their behavior phenotypes are normal. So this LX loci then is cloned or generated, and a congenic line is then generated to put this LX loci into the BN background, and that becomes a BNLX cup strain. It's a fully inbred strain, and this BNLX and SHR then are used to generate this HXB panel, and this work is done at Czech Republic. There are 30 inbred lines from these two parental strains, and there are many SNPs have been defined, using whole genome sequencing methods. Haplamaps are also being generated, and these are available from different sources. One of them is Gene Network. And many traits have been mapped using this panel. So because this is HHR, so many of the cardiovascular traits have been mapped, and limb development and malformation have been mapped. And there are also many behavioral traits being mapped, including anxiety-related traits, elevated plasmase, conditioned taste aversion, so on and so forth. So the other panel of rat RI panel is this RXFE and FXLE. The LE here is the Long Evans rat. Long Evans has an outbred population, but this Long Evans is inbred for over 53 generations before they were used as a parent strain of this panel. The Fisher 344 is the other panel is the other parental strain, and these two parental strains are then used to generate this RI panel. This panel is generated in Japan, and there was not a lot of mapping studies done using them, but they have been used to map many physiological traits like organ weights, blood chemistry, and there are also some behavioral traits being mapped using this panel. So the topic of today is a hybrid diversity panel. The way you put together a hybrid diversity panel is simple. You take many inbred strains, and you take the two RI panels in both the case of mice and rats. There are several RI panels you can use. So you put them together, and then you have this so-called hybrid diversity panel. The purpose of putting together this kind of hybrid panel is to increase the resolution of genetic mapping, because within this panel you have many different genomes, and with this different genomes you have many genetic diversity, and that is key to increase the resolution of the genetic mapping. Because these are all inbreds, so you still maintain the advantage of having the inbred strain, which is that this is a renewable resource. You can measure different phenotypes using different individuals, but these individuals all have the same genome. So it's a way to accumulate different types of data from the same genome. And also because the genomes are the same, so once the genotyping is done, you don't need to do the genotyping anymore. And the genetic mapping can also be conducted using the same method, such as you can do it on the gene network. So this is one of the early success stories of using the hybrid diversity panel. Again, this is in the mice. This is the HMDP. They were studying bone mineral density using the HMDP, and there were 3 QTLs achieved genome-wide significance. And one of them is at the beginning of the chromosome 12, and under this QTL, there is this SNP, this SNP over here, which is in close proximity to this gene ASXL2. They were later on, they were able to replicate this significant association in a human population. So what we are talking about today is the rat hybrid diversity panel, and there are some advantages of using rat as a model organism. To most of us in this community, where we use rats to study addiction, one of the main advantages of rat is the rich repertoire of its behavioral phenotypes. So drug self-administration is the most widely used model for addiction. And I searched many keywords such as cocaine self-administration, heroin self-administration, etc., by limiting these words to the title of the abstract. So it's more focused search. And then I added either rats or mice, as you can see, for almost any drug self-administration studies. there are over maybe tenfold, or sometimes in a few cases only fivefold more literature from rats than in mice. So these procedures are much better described in rats. Sorry, let me disconnect my phone. And we are not only interested in self-administration, we are also, many of us are also interested in other complicated behavior procedures, such as reinstatement, withdrawal, extinction, incubation, escalation, etc. And these procedures model different aspects of addiction behavior. And most of these behaviors paradigms are first established rats and are much better studied in rats as well. So that is one of the main reasons that many of us are using rats to do genetic studies of addiction-related phenotypes. There are some other aspects of rat that are useful, such as they are bigger than mice, for example. So, for example, in the brain, you can better delineate different brain regions. You can, for example, delineate the share of accumbens versus the core of accumbens, and you can still get enough tissue for doing transcriptome studies or epigenetic studies as well. So these are some of the advantages of using rats, but it's never a clear cut, because where rat is superior than other model organisms. So, for example, even though CRISPR-Cas9 has been applied to rats, the amount of genetically manipulated animals are far less in rats compared to those in mice. So hybrid rat diversity panel, and by now I think it's you can all easily put them together, which is just to collect a bunch of inbred rats and put these two recombinant inbred panels together. And this actually is indeed a international collaboration and an international effort, because these two inbred panels are generated in 2 different countries, and all these inbred rats are available in US. And this is a large effort led by Mindy Doinel at Medical College of Wisconsin, and her lab or her group is actually rederiving many of these inbred strains. So I think this is my last slide, and what we want to capture in the HRDP is a panel of rats that have genetic diversity and phenotypic diversity, and we want to use them over and over again so that we can accumulate many of the phenotypes, and we can pile on multi-omic studies, and we want to be able to study the effect of genetic background, and we want to be able to do high resolution genetic mapping, and we want to be able to do gene by environment interactions. I think that is where I'm going to stop. Okay, Laura, back to you.
I don't know if one of the two of you are going to talk about the status on the sequence of the rats, but I think it might be worth at least 30 seconds, How, to just comment that all of these HRDP rats are being sequenced or already have been sequenced.
Yeah, Laura is going to comment on that.
Okay.
I think I officially have my mute off and I'm ready to go. You can see my slides, right?
Yep.
Yeah.
All right, so thank you, Hal, for giving such a great introduction of what the HRDP is. And to be able to use the HRDP for individual studies, we also, it's necessary to have the resources that go along with it. So I am actually going to steal a little bit of an idea from Abe Palmer and this idea of ecosystems and how building these resources on these genetic populations can be used to create an entire community that can trade data back and forth and really build some synergies on who's collecting what. So on the left side of the graphic, I'm talking about the rat community. So there's the hybrid rat diversity, heterogeneous stock, selected lines, genetically modified, and then other general rat studies. So there's a lot of people using rats, and I just have named a number of populations. And by no means are some of these better than others, but I think they're all giving different aspects of genetics and traits and phenotypes and how to put them all together. So really taking advantage of all these resources will really build a much more in-depth picture. And so then what the genetic resources right now that are out there for the rat community include the genome, the transcriptome, RGD, which is the rat genome database, Phenogen Gene Network. These are all resources where we house and maintain and build tools to access some of this rat omics data and the methods. And so right now, as the HRDP is being developed, so Hao mentioned Mindy, and I'll talk a little bit more about her soon. So she's been about 3 years into her grant to rederive the HRDP to make it available to US researchers. So as Hao mentioned, one RI panel is from the Czech Republic and one RI panel is from Japan. And some of the inbred lines are available in the US and some of them have to be rederived. And so as we build up this community, we're starting to build up grants and people that are doing work on individual phenotypes. And again, because of this inbred nature, we can share phenotypic information and start thinking about how to build upon each one of those. And then specifically some of the HRDP resources that we're building up. So like we said, Mindy's building the rats. We have strain-specific genomes that are being generated. We also have strain and tissue-specific transcriptomes, and then lots of other omics databases, including some recently gathered proteomics data on the panel that we can use to really layer on to do analyses within this database. So these are the 4 resources that I'm just going to go over briefly today. So the live rats and rat tissues, like I said, are available through Mindy Dwinell and at RGD. Genome sequence information is how Chen has been putting that information together. And so we'll go into more detail about not just the sequence of this HRDP, but also this new version of the rat genome. Then the group I'm involved with in the University of Colorado, where we've been gathering really in-depth transcriptome information from brain, liver, heart, and kidney on a subset of this panel as they become available and making that available to the research public. And then the last one that we'll talk about is the Gene Network website that has phenotype data, has some of the RNA expression data from the point above, that protein and brain proteomics, and then tools for mapping in this population. So when we talk about the live rats and live tissues, again, this is a resource grant to rederive all 96 of these strains. And so this is courtesy of Mindy. And so what her deliverables, so this is the first place that you go if you want a live animal. This is the first place that you go if you would like a liver from a particular strain or a blood sample. And that's what Mindy's resource grant is all about. They're also doing some typical phenotyping, some basic physiological measures in these animals, and they can also be contracted out to do some more in-depth phenotyping. You can get more information by linking to the hybrid rat diversity panel through RGD and email them about your needs. And then this is the current availability on strains. And so you can see over here is our classic inbreds. And then these are the recombinant inbred that Mindy is in the process of rederiving. So I should have counted these before I started, but it looks like we're getting close to 30 RIs that she's rederived there at the Medical College of Wisconsin. And so she's slowly doing a few from the HRDP or from the HXB, interwoven with some from the RI panel from Japan. And then she has all of these standard inbreds live. So right now she's already built this data, started building up these colonies to make all of these strains available to the research public. And so I do encourage you to reach out to her, take advantage of some of her resources. One of the advantages of the HRDP is we can select maybe a set of 8 strains that are very representative of the HRDP that you could do something like a pilot study in to observe the genetic diversity among those strains, which would guide you to decide which of the 96 you would do further studies on, or if you would need them all and what kind of power you would need. So the genome sequence information, so this is what Rob was asking about. And it's ironic that we switched spots because this effort has been driven very strongly by Hao and his team have done a lot of work with this and coordinating the efforts to get a new rat genome, reference genome out there. So they've done a lot of sequencing. All of the original rat genome was derived from a brown Norway rat. Oops. And so they've done some sequencing using long read sequencing and some short read sequencing and different types of long read sequencing, and done it with the entire model organism community to really build a very strong database. And I've taken a look at some of this data, so it is available through NCBI with the reference genome, and then Ensemble has also released both a transcriptome and a genome through their rapid release. So it's not on their official website, but you can download it and get to it and get that information. And we've seen very marked improvements in the genome and how we can align reads and where we've seen translocations and things like that Hao and his group, and I'm being intentionally vague, and maybe we can ask Hao a bit more about this in the discussion section to get a lot more of his expertise. And like he said, the UC Santa Cruz genome browser is pending. So they're doing this detailed comparison. I do think a note is on its way to being published about this new genome, fixed a lot of large inversions, fixed sequencing errors and structural errors, and then got a big improvement in annotation accuracy when it comes to actually mapping those protein coding genes. And so we just recently did a relatively large RNA-seq data set and compared it to mapping from RN6 to mapping from RN7, both directly to the transcriptome and directly to the genome. And we got marked improvements in our alignment to the transcriptome. And our genome, where we really saw the big improvements is when we took those paired end reads and getting those paired end reads to align in a concordant manner. Right. So what that's telling us is that the previous genome had bits and pieces that were out of place, and we weren't able to align those to both ends of a paired end sequencing in a very, very accurate way. But with this new genome, we are able to do that. Ensemble and NCBI, this is all of the resources right now, just giving you a glance into getting to that new genome information. But on top of all the work that Hao's done with getting that new version of the rat genome pushed through, but both he and Mindy have been working on getting the full genome sequence of the HRDP. So not just markers, not just a set of SNPs everywhere, or SNPs every few megabases, but actually getting the full sequence of each one of these strains to really improve both our mapping and our ability to really dive deep into some of the QTLs that we identify. Sorry, I'm so sensitive here. All right, so when we move to the transcriptome, so that covers the genome information. So many of the HRDP sequences are available now, and they will all be available fairly soon. We do have marker data from the STAR Consortium in Europe that is available now and can be used immediately, but it will soon be replaced with this full gene sequence information. So when it comes to the transcriptome information, Phenogen and our group in Colorado has been collecting transcriptome data both on long RNAs and short RNAs, so protein coding and non-coding, and small things like microRNAs, and building up those databases for people to take advantage of. Like I said, we do both aliquot ribosomal RNA depleted total RNA, so really getting anything long that is in the lieu of RNA in the cell, and then also taking that smaller and doing it separately. It requires just slightly different library prep techniques and sequencing techniques. So we just separate by size and do it all. Right now, we have 58 strains that we've sequenced in the brain. So that's out of the 96. So we're expecting a new version here in the next couple of months that will include all 58. And then we've gone really deep and done biological replicates for each strain. So what biological replicates in an inbred population means is that we can start talking about start separating technical and environmental variation from genetic variation, right? So we can look at the between strain variation versus the within a strain variation to really talk about how much variation in this population is due to genetics. So we also have our major databases, unfortunately being a female myself, that are male rats. We are looking at the female rats and looking at different parts of the estrus cycle to make a plan going forward about how do we want to collect these females in our database to give us the maximum amount of useful information. Then we have brain, liver, heart is a descendant from or data that we've taken as part of the STAR consortium or the Eurotrans group that was originally in Europe. And then our newest addition is some kidney data that we also have on these animals. And what we do with the phenogen, we take the raw data, we do all the preliminary processing. So we try to make it as easy as possible for you to use, and you can download at any point along this little process. You can download at the preliminary data. We do some additional processing and additional analyses that are available. And finally, you can view this transcriptome data through a browser. So you can look at what we call wiggle plots, that it looks at the read coverage in a given area of the genome. You can download the data directly, or we have a whole other list of tools that can be used to analyze gene lists. This transcriptome data that we've generated is also available through Gene Network. So Gene Network has now added the hybrid rat diversity panel as one of its groups under the rat. And you can see the phenogen data is in there. So what that means is then you can use it, you can do your own mapping, you can look at correlations with some of the phenotypes that are on Gene Network as well. There is also other transcriptome data that's available from Gene Network. I think this is mostly array data. And then there's also this new set of brain proteome data that I'll talk a little bit more about. And so this was recently collected as part of a pilot grant through the OPAR session. And I am so sorry, I have all of the reference to that gene at a later, that grant, I'm sorry, I should have annotated better, but I have the name and the PI and stuff in a later slide. I apologize about that. But all of this data is now, so he measured 9,000, over 9,000 proteins, and that quantitative data is now available on Gene Network too, which is super exciting to layer this on top of. So now we have DNA level information, we have RNA level information, and now have added on that protein level data. Phenotype information. So we talked about genome and transcriptome, now transitioning to the phenotype data. Again, like we talked about this, the panel, the HRD panel in general is in its infancy, and we're just recently getting funded to look at a lot of phenotypes in it. So historically, we have some ethanol related phenotypes, but how and his group and his colleagues are in the process of collecting nicotine data and oxycodone data. And we're also collecting some oxycodone data in a slightly different paradigm in Colorado. And finally, we keep collecting alcohol and doing some other omics data through our center. And if I do a word cloud over what phenotype data there is right now, mostly it's from the HXB-BHX that's on Gene Network. There's about 200 different records. We have a lot of cholesterol and fructose and a lot of physiological measurements as long insulin and glucose and weight measurements in these animals already, because the panel was originally derived from that spontaneously hypertensive rat. So we know that there are some differences in cardiovascular traits. There's also some phenotype data on many of the inbred strains that are part of the HRDP, and that is available through the rat genome database. And so that was kind of thrown at you, all those resources. I'm going to go through a quick demo that is just meant to be a very superficial overview of how all of these data sets can be put together and what the advantages of this using an inbred panel are. So we're not going to talk a lot about the biological implications at this point, but I just want to give you kind of a big general overview. And so I'm going to use alcohol consumption as my example and talk about the initial studies we did that link DNA, RNA to phenotype, and then extending it to other tissues and validation and where this leads us with other omics data sets. So in the first one, we did what we call a genetical genomics Phenolic approach, which just means that we're combining DNA sequence information, brain RNA expression levels, and variation in voluntary alcohol consumption among these rats. And why I specifically put the map here is because I wanted to highlight one of the advantages of the Hrdp is that DNA sequence was that we used in this particular analysis was gathered by the STAR Consortium in Europe, whereas the brain RNA expression levels were gathered in as part of the Phenogen project in Colorado. And then the voluntary alcohol consumption was done by a group at the Scripps Institute in California. So one of the advantages of the inbred populations is that we're using the same genetic background, but we're accumulating data not only across generations, but also across laboratories. And so we can, we can layer that data on top of one another. And so the genetical genomics is just putting the expression, RNA expression, the phenotype data, and the DNA sequence together by looking at overlap of any of the two comparisons. And what we came up with doing that was a candidate co-expression module with both annotated and unannotated genes that then we could link to making hypotheses about biological function. And one of the things that I maybe glossed over is this was all done in brain. So we were looking at voluntary alcohol consumption, alcohol preference, and looked at brain RNA expression. So one of the things that we can do, again, with an inbred panel is extend it to other tissues. We can extend it to other phenotypes. And when we looked at the same type of analysis, but instead of using that brain co-expression data, we linked out to liver co-expression data. And the two modules that we found that were associated with our phenotypes were representing two different QTLs for this trait when we looked at the trait association with DNA. So really saying that there's probably contributions from multiple tissues to the genetic influence on this particular trait. And we were able to think more globally about the gut liver brain axis by being able to put in multiple tissues for the same phenotype. When we think about layering over phenotypes, one of the simple ones to think about here is, especially with our alcohol example, is that most of that first study was done on alcohol consumption. We also know a lot of information about alcohol metabolism in these strains. So we can also link the genetic influences on alcohol clearance and circulating acetate levels, and look for genetic overlaps between the three and see if there's any story that's emerging there. Finally, you can do validation now because we have an idea about what to target through this, and do a CRISPR-Cas9 knockout and gain a lot more information. So just like other studies in HS rats, we can also knock out genes in these inbred animals and choose a specific background to knock it out in based on what we already know about that background. And then finally, layering this other omics. So I'm so sorry, Xu Sheng, this, I think you're on the call. This is all of his data that we are presenting here on this proteomics data. And so what's one of the advantages of having this HRDP and on-gene network is we can put it all together. And what I did was took that brain proteomics data and I correlated it with my alcohol consumption measures that are also in that database, and got the top genes for their correlation with alcohol consumption, and identify things. And then also within Gene Network, you can link out to the Web Gestalt, which does that enrichment analysis for you. So we can start layering all of these different things on top of one another to get in more detailed picture on some things and a more broad picture on other things. So I just want to conclude real quick that hopefully we've explained what the HRDP is to you, what we think some of the advantages are, and there just with any panel, there's advantages and disadvantages, but what a few of the advantages of the HRDP is that we can parse that genetic variation out. We can map quantitative traits with both precision and accuracy. We can accumulate all of this data so we can thinking about systems genetics and transomics analysis. And then finally, like Hao mentioned, we can do these environment and exposure effects while capitalizing on this renewable genetic background. Right, and where do we go from here? And Rob, I had your back. I've got the diallelic cross in there. I didn't forget about you. I knew I had to say something. And so thinking about a diallelic cross is the F1 between any of these inbred strains. And so, oops, I'm so sorry. I am so sensitive here. And so like Rob mentioned, with 96 strains, I have 4,560 possibilities. And that's if I don't care who was the mother and who was the father. If I switch and do reciprocal breeding and switch my mother and father, then I have over 9,000 possible F1 combinations from this panel. So you're not limited necessarily to 96 inbred strains because you can also utilize these F1 populations. And for completeness, I've included a few really good references, many of them that Hao's been involved in on thinking about these diallel crosses. Some of the other things that we've been thinking about with the HRDP is single molecule RNA sequencing. So now that we have inbred strains, we can dig really deep and start thinking about what are the genetically driven differences in transcript structure. We are adding different omics data all the time. We can move on to this gene by environment drug interaction. One of the things that I'm excited about, I think both Hao and R1 in Colorado, is that we're looking at RNA expression both in a saline and an oxycodone exposed group. So we'll start being able to tease out predisposition and response to these environmental effects and differential response to some of these drugs. We can think about a gene by mutation effect. So oftentimes in our CRISPR studies, we take one inbred strain and mutate it and just study that one strain with the mutation, but we know that genetic background can have an influence of the penetrance of the effects of mutating this gene. So now we have a whole panel that we could think about knocking out a gene and really digging into those gene by gene interactions at that point. And then finally, my baby, if diallel cross is Rob's baby, then differential co-expression is my baby. So we can think about with these large panels in the context of networks of co-expression, what networks are activated or disrupted based on an exposure. And so we can get real in-depth into that information. So there's lots of different places we can go from here, but I just wanted to give you a little taste of some of the things that have been on our mind as a group in the P30 and the Phenogen project. But obviously we think there's lots of different ways to go. And then finally, we just wanted, both Hao and I wanted to acknowledge the grants that have supported us to gather this HRDP information, the community of people that are working on the HRDP and trying to build that up and building these databases that we can start thinking about PHWAS and transomics and those sorts of things. And then of course, everyone in our respective labs. So I think that puts us at about 53 minutes. So I think we can turn it over for discussion and questions.
Yeah, thanks very much. That was great. I just wanted to initially just pile on with your next to the last slide about what one can do. Many of us are interested in mapping gene variants that control traits of interest, whether it's a substance use disorder or initial response or relapse or an operant conditioning trait. But the next stage of this work should be to see whether we can assemble strong causal models, take our variants, take the environmental perturbations, and make predictions with reasonably high receiver operating characteristics and say, okay, if I construct that mouse, that F1, if I take those two parents and in 3 months I have progeny F1s, which have a completely defined genome, can I accurately predict what the best environmental perturbation will be or intervention to either make them healthy or make them not healthy, preferably to make them healthy. After, say, an exposure to 5 doses of oxycodone, which they might get, which a human might get if they go into a hospital with a broken leg or something. So you want to know, can we actually do accurate health interventions in rats first before we try to do this in humans? And because you don't have the N of 1 problem that we have with humans, you can do what Hao and Laura mentioned. You can do the G by E until you get the right E to be useful to the organism in terms of its long-term health outcomes. And I frankly don't think we can do precision medicine without experimental platforms of this type. And as Laura said, there's close to 10,000 F1s that are completely defined and where we'll have telomere to telomere assemblies over the next few years. So you won't want for anything in terms of the genetic basis other than a few rare de novo variants. And those actually may come in handy. They've been very handy for some of the work that we've been doing with the BXDs. So I'll just stop there. I didn't see any questions, or Stephen, you had some questions and I tried to field them during the talk, but if you have any others, fire away.
No, I'm good. Things popped into my head and I just typed them into the chat.
Well, and I did want to, one other thing I wanted to chime in, because I did see a lot of people on this webinar that traditionally use HS rats. And I want to be very, very clear that I don't think it's an either or, that I think there's a lot of benefits from adding results from the different populations that are really exciting. So for instance, the HRDP, although we're going to get more precise by using a hybrid panel, we're not going to get to the precision that an HS population gets to. But what we could do is put those, we could, join the human bandwagon, have a test and a validation set. We could make predictions off of the HS and then test them, because we know those genetic backgrounds in the inbreds and we can control that environmental exposure. So they can almost be as a way to follow up some of those HS results and vice versa.
Yeah, and all but one of the HS founders, I think, is part of the HRDP. Is that right, Hal? Or do we have them all kind of, sort of?
It's kind of sort of, because many of these HS founders are no longer available as live rats, not even cryopreserved. They try to, I think Mindy's group tried to what's the word for that? But it was difficult.
But we'll have basically the same kind of variation that you see in the HS. We'll be segregating in the HRDP and vice versa. So you can, I think Laura's exactly right. It should not be a zero sum. It should be, the hardest thing to predict would be the behavior of an HS rat in response to a substance, an injection. But you should be able to do that in combination with the HRDP. You can say, okay, I have HS data for 500 rats that were treated like so, and I have a beautiful locus, and my prediction is that these HRDP F1s should have this outcome, and these HRDP should have that outcome. And now you can do 10 and 10, a kind of classic t-test if you want, and you can just test that one locus. So, and you can, everything else is now reasonably, well, is potentially definable. So it's a potentially very powerful design and kind of a next generation wave of predictive biology rather than just essentially, you know, no matter how mechanistic we get, it's description right now. We're describing fundamental biological processes, but we need to be able to predict and understand biological processes.
Yeah, I think one more fundamental question is what are the genetic diversities that are present between the HS and the HRDP, like you were saying, Rob? So all the founders, even though they are not available as live rats, that DNA have been sequenced. So we should be able to just compare the genetic variants between these two populations and see how much overlap there is. I suspect that the overlap is quite large.
We were very deliberate when we were picking inbred strains for this panel too. We looked at phylogenetic trees. We tried to get every branch of the we did get every branch of the phylogenetic tree represented. So although we don't have some of the HS founders, we have strains that are somewhat similar genetically. And I do, Shusheng, again, I am so sorry I had your name later, not earlier. But one of the things that I didn't have time to do was really integrate that data a little bit better. So exactly like Rob was talking about, this causality effect, that we can start playing with our statistical models. We can start playing with our Bayesian networks and causal inference and structural equation models and stuff and things like that with this layered omics data to try to see if we can direct those edges, if we can talk about causes and effects when it comes to both genetics and environmental perturbations.